All right, welcome y'all to the first Exam 1 review session. It's going to go from 6 to 7 tonight, and then tomorrow will be a completely different review session from, I believe it's 10 to 11 in the morning. So with these reviews, how they work, sometimes I'm going to use documents that have already been posted on the review resources. I know some people, may have already tried to complete them. So normally the day of or the day before, I'll post something kind of extra to do. That way you're not just sitting through something you've already done. I'll try to get things out in advance. I was telling some of y'all earlier, I get married in 2 weeks and 58 minutes. And so there's just a lot going on. So I'm sorry for the last minute post of the documents, but we're gonna start with, hold on, how this is gonna go. So each review is normally gonna focus on like one big topic, and then we'll cover some of like the smaller topics. So like today, the big focus is gene expression and horizontal gene transfer, because that's where so many people lose points on the exam. So we'll do that, and then if we have time, we'll kind of maybe start talking about cells, or like a smaller topic like the scientists. And then tomorrow, the big topic is going to be viruses, because that's the other one that people tend to get confused on. And then we'll do another smaller topic with any remaining time. Now, these are meant to be interactive. Some of you have to deal with me during the school week. And it tends to be in lecture me babbling most of the time. And so with these, I will be the one mainly talking, but I'm going to ask a lot of questions. I do this in my classes, in-person classes as well. So please feel free to answer the questions in the chat or come off the mic. If you have questions as we go, do not hesitate to ask. This is your time. So we're going to start with the gene expression word bank. And as you can see, you're going to want to get a blank sheet of paper and divide it into quarters or quadrants. And then you're going to label each one as follows. And then we're going to talk together about putting all of these words where they go. And this is just going to be a jumping off point to talk about these big processes. Because can everybody else still hear me? Okay, hold on 1 second. Of course, I can't type when I'm like in the moment. Okay, so you may have noticed that we're talking about a lot of processes for this exam, including transcription, translation, transduction, transformation, right? Lots of words that sound really similar. And so on this exam, people sometimes read too fast, or they just kind of get these words confused because they sound so similar. So the point of this activity is to help you separate out what kind of ideas go with which big topic. Okay, so what you're going to want to do is this. Now, don't put the word text there. Okay, that's where I'm going to be typing in the words. So on your own, I'm going to go ahead and mute myself for a minute so that I'm not causing any distracting noises. Write these 4 words down. And if you're not a quadrant person, that's okay. Just kind of list them out and then use the words from this word bank to fill in. Okay, codon. What process are we concerned about? Codons. Okay, dipole 3. Okay, well, what process is DNA polymerase 3 involved in? So take these words and put them in the respective box or category. Now, there might be 1 or 2 where you're like, oh, it could be this or this, and that's okay as long as you can justify why. So I'm going to give you 2 minutes at 6:07. I'm going to come back on and we're going to go through it together. But try your best to just use your brain. Try not to use your notes for this review. All right? Take one more minute and just do your best. I suggest put every word somewhere because getting it wrong is actually going to help it stick. So every word, take a guess if you don't know. All right, make your final selections. And I'm going to move the word bank to my other screen. Hold on one second. I'm on my fiance's setup, so I'm a little, oh, this is going to be hard with only 2 screens. All right, I'm going to do my best. I'm going to have to kind of go back and forth between screens. Okay. Yes. Yes. Fluffy bunny, you are correct. Okazaki fragments are for the lagging strand. Yep. Okay, so let's kind of start on the left-hand column and work our way down. So in the chat, y'all don't be shy. Where should we put the word codon? Which process? DNA replication, transcription, translation, or horizontal gene transfer? Where would codon go? Excellent. Yes, translation. Can anybody tell me what is a codon and where do you find it? Hey, yes, good detail as well. Excellent. Yes, the 3 letters. And remember, those letters are nucleotides. And so I'm going to abbreviate y'all, but a codon is 3 nucleotides found on messenger RNA. And that's really important, okay? And as I talk about things, of course, feel free to add your own notes. Because with translation, okay, the goal is to create protein, okay, which is made up of amino acids. We need to know how to go between the RNA nucleotides. How do we read that RNA to figure out how to make a protein? Because, right, RNA and protein are 2 very different things. And so we're going to read the RNA, specifically the messenger RNA, in sets of 3 nucleotides. And so I remember that codons go with mRNA because M stands for message. So the mRNA is literally a coded message that tells us how to build a protein. Excellent job. All right, working our way down the list, DNA polymerase 3, where does that go? Yeah, good job. Yes, so DNA polymerase 3, and I don't have all of the key players on here. So if I were you, I would write dpol 3 and dpol 1, right? And if you want, on your exam, I do spell it out to say DNA polymerase. And so remember, y'all, the name gives everything away. DNA polymerases make DNA. The only process that makes DNA in the cell is DNA replication. So remember though, y'all, because they're DNA polymerases, both of them can build that strand of nucleotides to make the DNA. But remember, they do kind of different things as well, okay? So remember, DNA polymerase 3, in my stories and the videos and stuff, I say that's the third born, the baby of the family. That's the key player. That's the main one that's building that strand of nucleotides. But it's a little high maintenance, right? It doesn't just start building DNA on a whim. It kind of needs to be primed. It needs to be, you know, started. And so DNA polymerase 3, remember, it cannot start from scratch, and it can only add nucleotides to a 3' end. Okay, and so that's what dipole 3 does. It builds that chain of nucleotides. But dipole 1, remember, is the big sister. It's like cleaning up the mess of dipole 3. And so dipole 1 has one really important job during DNA replication. Does anybody remember what it is?
Can you repeat the question for me? I wasn't paying attention.
Sure. What does DNA polymerase 1 do?
Does it fill the gap, but not the little fragments that ligase fills?
Yeah. Well, hold on a second. Not quite. I was trying to listen to all the things, and I'm not sure I heard you entirely correctly. It is going to, so remember, DNA polymerase 3, it is high maintenance. Remember, it needs help getting started. And so one thing it does, excuse me, is it needs a primer to get started, an RNA primer. And then the dipole 3 can attach to that primer and add nucleotides. But we can't have RNA in the DNA. And so dipole 1 has to come in and remove that RNA primer, and then ligase comes in and fills in the gap. Okay, excuse me. And then yes, dipole 1 will then also fill in that gap laid by removing the primer. Good, good, good. Y'all definitely, I promise, you're gonna want to know what the key players do for DNA replication. Okay, let me get back to my list here. All right, next up is conjugation. Where does that go? Excellent. Horizontal gene transfer. Where is that? Right over there. Okay, so there's another word in that list that is associated with conjugation. So let's go ahead and address that one now so that we can kind of be pairing these processes together. Excellent. The pilus. So remember, I compare the pilus to like a fishing line. So think of it as being like two neighboring cells just hanging out, minding their own business. One of them has the F plasmid, and so it can make a pilus. And so it's like, hey, I don't know if you can see my video or not, but I tend to be kind of like a goober. This one's like, hey, I can make a pilus. You should be able to as well. So it casts out the pilus like a fishing line, and it connects to the other cell that was just minding its poor business, and it brings it close together, much like you do if you catch a fish. And then once they're together, that pilus becomes a mating bridge. So it kind of forms a hollow bridge, and then they share that fertility plasmid. So yes, so let's put some other words on here that you should remember with conjugation. The F plasmid, which stands for fertility plasmid, and then mating bridge. I think that should be, that's the gist of it. Let me make that longer. There we go. So good. So remember, with this form of horizontal gene transfer, cell-to-cell contact is going to be required. Okay. Polypeptide chain. Where might that one go? Good. Translation. Yes. And here's why. I'm trying to go back and forth on too many screens. Does anybody know like what a polypeptide chain actually is? Yeah, Christina, you're on the right track. It's a chain of amino acids. Yeah, so it's basically a pre-protein, because in order to be called, exactly, good, Katie, yeah, because in order to be a protein, those amino acids have to fold into that specific 3D shape. But when the protein is being built during translation, it's kind of like an assembly line. It's like, all right, amino acid 1, 2, 3, 4. And they connect them together, but they just form a chain. There's no 3D shape yet. And so because it's not folded, it's not considered a protein because it's not functional. It can't do anything. Once the ribosome and everything dissociates after translation and that big old polypeptide chain is released, it will then fold into its shape. It can then do its job, so it's then earned the right to be called a protein. So yeah, so a polypeptide chain is what's actually made during translation. Good, excellent job, y'all. So Katie, it's going to depend on the protein. That's a really good question. So all polypeptide chains will eventually fold to the tertiary level. And for some proteins that are small, like that's it. But other proteins that have more complex jobs, there's going to be multiple polypeptide chains that have to work together. And that's going to be what gets it that quaternary structure. So it's going to kind of depend on which protein we're talking about, if it goes to the quaternary stage or not. All right, so let's look at our list. Next is bidirectional. Where do you think bidirectional goes? DNA replication, good. So remember, y'all, and let me see if this word is on here. I don't think so. Okay. So remember, the 2 parent strands of DNA, right, are going to be separated. And that happens, and let's go ahead and add this word, to the origin of replication. That's what we call the starting point of DNA replication. And at that origin of replication is where the DNA strands start to separate. And I can't really mirror this with my hands. It does so in such a way that it forms a bubble, that replication bubble, right? And it's in that bubble that the new strands of nucleotides are made. And because this has to be a really quick process, within that bubble, DNA is being copied to the left and to the right at the same time. It's just a time-saving mechanism, okay? So that's what we mean by bidirectional. Both halves of that replication bubble are being copied simultaneously. All right, so we kind of just answered where this next one should probably go, but let's go ahead and put it. Protein synthesis. So remember, y'all, protein synthesis is the act of synthesizing or making proteins. So which of these has the goal of making protein? Yeah, excellent. All right, I grabbed the wrong one. Good. So y'all, what you really want to do, if you can't already, and you've got a couple of days before the exams is due, you're going to want to be able to like take this word list and fill this out without hesitation. Anytime you see the word translation, you need to go, protein synthesis. We're starting with RNA, we're making protein, okay? Because as I mentioned, a lot of these words sound really similar, okay? And so you don't want to hesitate when it comes to, wait, what is that trans sounding word again? Because on the exam, all these words are going to be familiar, but you have to know quickly transcription versus translation. All right, good. So next up is going to be anti-parallel. Where does that go? Good. And we're going to go ahead and to save a little bit of time, let me see. I don't remember every word I put on here. All right, so the 2 strands of DNA, the 2 strands of nucleotides that make up DNA are anti-parallel and what's the other word? It's the science word for base pairing.
Complementary?
Yes, good job. Complementary. So then what does it mean for them to be anti-parallel?
So like 5' to 3' and then 3' to 5'.
Exactly. They run in opposite directions. The 2 strands are linear, so they're parallel. And then yes, they just move in opposite directions. Excellent. Good job. And so that's going to be really important if you go back to the videos or the PowerPoints. For those in person, we did it together in class. When I give you a strand of DNA, you should be able to give me the other strand that makes up DNA, because the two strands are anti-parallel and complementary. So that's a really good application question. All right, let's see what's next. Ooh, finally, okay. RNA polymerase. Where does RNA polymerase go? That's what RPOL is. Good. All right, Taylor, awesome. What else goes with transcription? Let's kind of talk about each process one at a time, just in case. I think it might just be easier to keep track of all that. So RNA polymerase is our key player for transcription. What is the goal of transcription? What are we making? We are making messenger RNA, but I'm going to take that one step further. We're making RNA in general. So we call this RNA synthesis. So remember, yes, you're absolutely right. We make messenger RNA, but the cell also makes tRNA and ribosomal RNA. All of them are made during transcription. When we move on to translation, we're mainly using the messenger RNA. Good. All right. And then Taylor, I think you put in there promoter. Good. What is the promoter? That's going to be good. That's going to be our start site. And remember, it's on DNA, because during transcription, I'm going to kind of just be adding things as I talk. With transcription, that's where we're starting with DNA and we're making RNA. Of course, I don't have the arrow symbol, but I can get it. All right, and so all of this begins at the promoter. It's a piece of, it's a piece of a sequence of DNA that says, hey, RNA polymerase, you need to get right here because we need to make some RNA. So RNA polymerase is going to bind to that DNA sequence, that promoter, and then it's going to start building a strand of RNA, okay? And so what you need to remember, though, is that RNA is single-stranded, okay? So during transcription, there's both strands of DNA. They're going to separate temporarily, okay? Not permanently like they do in DNA replication, temporarily. And so they're going to temporarily separate, and only one strand is going to be used as a template, okay? And it's going to be a template that's used to make an anti-parallel and complementary strand of RNA. Now, we're concerned about messenger RNA, right, as we move on to translation. What directionality does messenger RNA always have? There we go, good. 5' to 3', okay? And that would be the case for all RNA, okay? Because remember, all of them are made during this process. But because we're gonna use messenger RNA later, I'm gonna focus on that. So good, it's made 5' to 3', perfect. Okay, is there anything else in here that we need to put for transcription? Oh yeah, the bases. What bases make up RNA? I already put it on there. Yeah, A, U, G, and C. So I'm going to go ahead and I'm going to bold that just to remind you. Okay, you will not find uracil in DNA. All right, let's go ahead and kind of backtrack and let's go back to DNA replication. What are some other words that belong in there? Some of our key players. And you can even throw out things that aren't on this list. Think about some of our key players, for instance. Good, topoisomerase. Remember, that's the enzyme that relieves supercoiling, because as those two strands of DNA get separated, it's going to cause the unseparated portion to get wound up so tight that it can break, and then the organism's not going to survive. So topoisomerase is preventing it from getting wound up too tight. Excellent, Emily. Yes, semi-conservative. Remember, that just means that at the end of DNA replication, we have two pieces of DNA, okay? And we say they're semi-conservative because each of them, if we look at their double strands, one has they both have an original parent strand, and they also have a newly made strand. So because each of those DNA molecules, each of them has an original strand, that's where the conserved part comes in, okay? So it is a semi-conservative process. Excellent. All right, what else? What else can we put in there? What bases do we use during DNA replication? Yes, good. And I'm going to bold that thymine, because again, that's one of the big differences between DNA and RNA. So remember, that is an important thing to remember. DNA is double-stranded, RNA is single-stranded. DNA has thymine, RNA has uracil. DNA has a specific shape. It is a double-stranded helix. Messenger RNA, transfer RNA, and ribosomal RNA have their own shapes. So there's not just one uniform RNA shape. Excellent. All right, I think there's a couple more words on there that we could use. I'm going to go ahead and type some of them out. Feel free to put them in the chat. I'm going to move these boxes up. I didn't give myself enough room here. All right, so we've also got ligase. Oh, I don't know. Why is that still bold? Ligase, primase. Yes, good. Single-stranded binding proteins, and that is one thing I do abbreviate on the exam, SSBP. Good. Let's see, we've talked about dipole 1, dipole 3. Oh, helicase. I think that's it. 1, 2, 3, 4, 5, 6. Nope, we're missing one. Oh, we already got 2 isomers. Nope, we're good. Okay, good. Make sure, y'all, you know your key players. All right, then the other thing is we talk about in the videos and in class, the leading strand versus the lagging strand. So because DNA polymerase 3 can only add nucleotides to a 3' end, one of the strands of nucleotides, once dipole 3 starts its job, it just keeps on going. It's made continuously in the direction of the replication fork. Easy peasy. But on that other strand, it makes it in pieces, and those pieces are called Okazaki fragments. Okay, and so it has to build that other strand of nucleotides in pieces. So this is where primase, dipole 1, dipole 3, ligase are working in overdrive because they're constantly having to, build the fragments or connect the fragments or whatever their specific role is going to be. All right, I think we got all of the DNA replication ones. Any questions so far, y'all, about any of these processes? Please, please don't be shy. All right, let's move over to translation. All right, Griffith is actually going to go in horizontal gene transfer, so we're going to circle back to him. So with translation, remember our goal here, let me scoot it over so I can type some more. With translation, we're focusing on mRNA to protein. Okay, now, please make sure you understand that the messenger RNA is not becoming protein, like it's not a magic trick, it's not changing into it, but we're using messenger RNA to make protein. And proteins are made up of amino acids. Okay, these are things we already talked about. All right, what are some other words that go under translation? Good. So I'll put our key players here. And I know not all of them are on there, but yes, we've already said messenger RNA is going to be the directions, okay? And then yes, transfer RNA, and then the ribosome. And the ribosome has 2 pieces, okay? It's the large and small subunits. Good, exactly, y'all. All right, so here's how I think of these key players. The messenger RNA is going to be the directions. We need methionine first. I need proline second. I need serine third. Okay, it's basically barking out the orders. The transfer RNA is the little worker that's like, oh, okay, I'll go get methionine. All right, here it is. Who's next? Okay, I'll go grab proline, and it brings it there. And then it grabs serine and brings it, to the ribosome. So the transfer RNA is the one actually going out to the cell, grabbing the required amino acid and bringing it back. It's doing all the work. The ribosome is like the factory where this is happening. It's like the little location, okay? Because remember, ribosomes are the protein-making machines of the cell, okay? So 3 key players there. And I think we've, the other one. Okay, so we said what a codon is, 3 nucleotides found on messenger RNA. And so then we also have anticodons, which are 3 nucleotides found on transfer RNA. If we have time at the end, I have an extra little worksheet we can do to make sure we're good with this process. And if not, I can still post it tomorrow after the review if we don't get to it. Just a good practice. We do it in the content videos, and I did it in class, where we start with DNA, we figure out what the RNA is, and then we figure out what the amino acids are. So you definitely want to make sure you know how to do that. Okay, so let's wrap up this word bank with our horizontal gene transfer. What is, I guess I can just, what's another form of horizontal gene transfer? Good, transformation. So remember, y'all, with transformation, this is just a single cell kind of minding its own business out there, and it encounters a piece of DNA. Normally, it's a fragment of DNA, normally from a dead cell that kind of, when it died, sometimes the cells like literally blow up. And so then all of the stuff from inside the cell gets released into the environment, including little bits of DNA. So this is where a cell, if it is considered a competent cell, okay, can pick up pieces of DNA from the environment. Okay? No other cell is needed. Okay? There's no cell-to-cell contact, nothing like that. Okay? It's just a cell minding its own business. It's like, oh, there's a piece of DNA. I think I'll take it in and see if I can use it. And if it can, it does. If it can't, it just kicks it back out. Okay? And so remember, only some cells can do this. It's not a normal phenomenon because DNA is negatively charged. Even little pieces of DNA are pretty big, okay, compared to a cell. And so these charged large molecules aren't compatible with the plasma membrane of the cell. And so very few cells can naturally pick up such a bulky charged molecule. All right, and then our last one is going to be what? Yes, transduction. And so remember, this is when a bacteriophage, which is a virus that infects bacteria, this is an accidental process. So if you've watched the virus videos, then you'll know that with bacteriophage, they take over a cell, just like animal viruses. They make more copies of their genetic material. They make lots of copies of viral proteins. That's during the biosynthesis stage. And then during maturation, they come together. All the viral proteins come together. They surround that nucleic acid. They make intact virus particles, and then they get out. And so sometimes during that process of spontaneous assembly, the bacteriophage accidentally grabs bacterial DNA. Capitalized, but that's okay. Not viral DNA. Man, my typing is not on point today. What am I typing? There we go. Okay. And so tomorrow morning, and I'll be recording that one as well, I'll be going through all the virus stuff. So we're going to touch on this one again. But if I ask you about horizontal gene transfer and bacteriophage, just know right off the bat, that's got to be transduction. It's the only one that involves a bacteriophage. All right, I think we got through all of these words. Are there any questions? Okay, so let me go ahead and get the new worksheet up that I posted right before the review. Give me 1 second. Take a peek at that beautiful castle right there. Anybody's ever been to Germany? That is the Neuschwanstein Castle. It's stunning. Oh, looks like I closed it. I went to Germany. I'm going to tell you all this just while I get my document pulled up. I went to Germany back in 2019, summer of 2019, before everything went crazy. Hold on 1 second, y'all. It's telling me the file has moved, and it has not moved because I created it 30 minutes ago. But yeah, so I went up, and as you can see, that castle is pretty high up. And I knew that because I've seen the pictures. But what I didn't know was it was like 1.5 miles up the side of a mountain. Quite a hike. There we go. Now it pulled up. But totally worth it. Absolutely stunning castle. Okay. Hold on, I heard somebody. I did not take this picture. This one is a professional, but I have some not quite that quality, but pretty close, I'll say. But what they do to reward you for making it to the top, on the way back down, there's a German individual that is making, like on the spot, donuts, fresh donuts. And so you get fresh donuts and oh, what do they call it? The spiced wine. Can't think of it right now, but you get that on the way back down, and it was like totally worth it. It was the best, one of the best memories of my life. Okay. All right, so what I'm going to let you all do here is I'm going to give you about 3 minutes where I'm just going to be chilling here. I'm going to go on mute, and I want you to go through, and I don't know why I actually called it a true false. Each of these statements is false. And so what I want you to do is change it to make it a true statement. And for most of these, you can change just one word and it'll make it a true statement, okay? And there are different ways you can edit it. So just pick one for now and get as many as you can do in 4 minutes. So at 6:43, I'll come back on. Try not to use your notes. Okay. I've seen a couple people jump on, so if you've been here, ignore my voice and keep doing what you're doing. What we're doing is we're editing each of these statements to make them true. And normally it's just one word that you need to change. And we want to make these true statements. I'm gonna give you about 2 more minutes and then we'll start going over the answers. All right, y'all. So let's go ahead and take it from the top. So what is one way that we can edit that first sentence to make that statement true? What's one word we could change?
Eukaryote.
Yeah, there's a couple of different ways, right? So if we wanted to leave transcription occurs in the nucleus, then we would need to change this, right, to eukaryotic. Transcription occurs in the nucleus of eukaryotic organisms. That is absolutely one way we could fix it. If we wanted to keep talking about prokaryotes, we could then say transcription occurs in the cytoplasm of prokaryotic organisms. And here's really what you have to remember. And I at least tell my in-person students this all the time. Let's see if I can do this without making them document mad at me. You need to always tell yourself, when you see transcription, you need to say, okay, the goal of this is taking DNA to make RNA. So if we're starting with DNA, where is the DNA, right? And so for eukaryotes, DNA is in the nucleus. So transcription has to happen in the nucleus because that's where the starting material is. But prokaryotes don't have a nucleus, right? So for prokaryotes, the DNA is just inside the cell. We call the inside the cytoplasm. Okay? So good job with that one, y'all. All right, number 2, DNA is used as a template for RNA synthesis during translation. What's the problem there? Good, exactly. DNA is used as a template for RNA synthesis during transcription. Yep, that should say transcription. Excellent. So again, remember, DNA to RNA is the goal. All right, number 3, RNA polymerase binds the start codon to initiate translation. So this one you can actually, just changing one word, not as possible. So how could you edit this sentence to make it true? I'm gonna wait just a second to see if people are typing. Good, Colette. That is one thing you could definitely change. So let me kind of bold this so I can show you the changes. So yes, RNA polymerase is the key player for transcription. So that's good. That's one change. But we would also need to change the start site, right? Because where does transcription start at? As Taylor says in the chat, the promoter, okay? So RNA polymerase binds the promoter to initiate transcription. Good. Yeah, that was one that's a little more tricky, not just one word. All right. Ligase is used during DNA replication to create a RNA primer. What's the problem with that one? Good, Taylor, it's like you know what I'm gonna ask. Good. Ligase does not make a primer. Rather, that should be primase. That's why it's called primase. So remember, y'all, when you're studying this, try not to just memorize a bunch because your head might explode. Look at ASE, say, okay, cool, that's an enzyme, and then look at everything before it. It's called primase because it lays down the primer. Ligase ligates things. Ligate means to connect, and that's what it does. Helicase separates the helix, why it's called helicase. Okay, so try to give your brain a little break. All right. Oh, sorry, y'all. Number 5 should not be on there. My apologies. We are not doing RNA processing. All right. Number 6, topoisomerase is used to separate the parent strands during DNA replication. What's the problemo there? Not quite. It keeps them separate. Good. Yeah. So helicase actually separates them, but they're going to want to go back together, right? Because they're complementary and anti-parallel. They want to be besties. So yeah, helicase is going to separate them. The single-stranded binding proteins keep them separate. Yep. And then topoisomerase relieves the supercoiling. Good. And so, yeah, if we wanted to, as Colette here is saying, we could have edited instead, we could have edited this part. We could have said topoisomerase relieves supercoiling during DNA replication. That is also an excellent revision. That's why I like assigning these. I used to assign them as like homework, but y'all now have a lot to do in this class as it is. But I love to see how people's like brain works and what they choose to change. Okay. Here's a picky question. This would be considered a hard question on the exam. During the initiation of translation, messenger RNA first binds to the large ribosomal subunit. What's wrong with that? Good. It binds to the small ribosomal subunit. Yep. So remember, during initiation, the point is to get all the key players to the start site. So in this case, we need messenger RNA, transfer RNA, and the ribosome, the intact ribosome. But the ribosome doesn't hang out intact in the cell. It only comes together when it needs to make a protein. And so the messenger RNA first binds to the small subunit, and they kind of lock each other into place. Then the initiator transfer RNA will bind to the start codon. What's the start codon? Good, AUG, excellent. So then that initiator binds to the start codon and it's carrying the amino acid methionine, okay? So yeah, just be careful there. The start codon, remember codon is a set of 3 nucleotides, that's the AUG. The amino acid it's carrying is methionine. And then once all those are in place, the large subunit comes in and kind of finishes the process. So the large subunit is the last thing to come together. Good job, y'all. I thought that would be a lot trickier. Okay, number 8. Translation occurs in the nucleus of eukaryotic organisms. What's wrong with that? All right, so yeah, so we could change that. It makes it a little redundant to number 1, but it's absolutely true. Yep, transcription occurs in the nucleus. Or what you could have changed is this word. Translation occurs in the cytoplasm of eukaryotic organisms. Translation also occurs in the cytoplasm of prokaryotic organisms as well. Because exactly, translation, the goal is to make proteins. So we start with RNA and we end with protein. Well, in order to make proteins, we need the ribosomes, right? Ribosomes aren't in the nucleus. They're out in the cytoplasm. They're outside of the nucleus. All right, y'all are doing awesome. Okay, number 9, transfer RNA molecules. Transfer nucleotides to the growing polypeptide chain. Oops. Hold on, sorry, y'all. Let me figure out. I have to get used to his setup here. All right, let me see, what did y'all say? Right, so nucleotides is wrong. That's absolutely right. And what should it be? Oh, good, y'all got it. Sorry, I didn't even see that. Yes, transfer RNAs, are going to transfer amino acids, okay? Because remember, we're trying to build a protein during translation. Proteins are made up of amino acids. So something has to go and get those amino acids and bring them to the ribosome so that they can be connected together. And so that's what the T stands for, transfer. It's transferring amino acids to the ribosome so they can be brought together. Good, that was a pretty detailed one too. All right, last but not least, DNA replication is said to be a conservative process. What's wrong with that? Where's this file? Good, it's a semi-conservative process. Yep, and so remember, we mentioned this earlier, DNA replication starts, uh-oh, my computer's about to die. Hold on 1 second. Stay alive, computer, stay alive. Oh, it's not cooperating. Hold on, y'all. Holy moly, he's got all the outlets. Hold on. Let's see if this works. Ha, yeah. Phew. And yeah, for my online students, I'm this much of a hot mess in class too. Okay, so good. Remember, we say it's semi-conservative because when looking at the 2 DNA molecules that are made, each has an original parent strand and each has a newly made strand. Good job, y'all did really well on that. All right, so let's wrap up this conversation by going quickly over cells, okay? And we can come back to this more tomorrow. We don't spend quite as much time on viruses as I do with this. Was that a bit of Hamilton? Was what a bit of Hamilton?
No, I thought for a second you were singing a song from Hamilton.
Oh,
Stay alive.
Oh no. I try not to sing to people because eardrums start to bleed. All right, so if y'all can kind of navigate back to those, the resources in the exam review resources was the prokaryotic and eukaryotic Venn diagram. And I'm going to be posting some new documents for tomorrow's review session. So if you've already done them, it'll be a bunch of new stuff. But this is really important because this is something that a lot of students get confused on the exam is what's bacterial, what's prokaryotic, what's eukaryotic, but what's both, okay? And so looking at this Venn diagram, let me get my mouse to cooperate. I want you to go ahead and put the structures that only eukaryotes have over here, structures only prokaryotes have over here, and then what structures both can have, okay? And I know we're down to 4 minutes, so just take about 1 minute, and as always, put everything somewhere, okay? And try not to use your notes. 1 minute. All right, it's been 1 minute, so I'm gonna go through some of these real quick. So remember, what makes a eukaryote a eukaryote? 2 things, nucleus, organelles. Remember, organelles are like little organs, okay? All the little individual things that help a eukaryote work and survive. So the nucleus should go under eukaryote, and then so should any organelles. And so if you look, trying to get this in here, now it's going way too slow. Why is Word being cuckoo birds? Okay, well, I'll just tell you. So the mitochondria, the lysosome, and the chloroplast are all organelles. So for a eukaryote, you should have nucleus, mitochondria, lysosome, and chloroplast. Okay? Let me highlight those so I can at least keep track of ones that I've said, since it's not letting me do a text box. So these are all eukaryotes that I'm highlighting in yellow. Eukaryotes only, I should say. Nope, prokaryotes do not have a mitochondria because that's an organelle. All right, so some structures that only prokaryotes are going to have, I'm going to highlight those in green. That's going to be a capsule. Remember, that's the sugary, sticky outer layer that our immune system, some of our immune cells, have a hard time grabbing a hold of. So capsules are kind of like, we call them virulence factors. It helps make organisms more pathogenic because they evade our immune system easier. So capsules are going to be on a prokaryote. And then the other one unique to prokaryotes is the fimbriae. Remember, those are little small hair-like structures that help bacteria stick to things. All right, so what do both have? Or I should say, what can both have? Okay, so let's start with actually, let's do an all statement. All cells, no matter if they're prokaryotic or eukaryotic, all cells are going to have ribosomes. All cells, exactly, are going to have a chromosome, because remember, that's just DNA. All cells are going to have a plasma membrane. Because remember, that's the border of a cell. Okay, so that's all. Okay, oh, one more. All cells have cytosol. That's the jelly-like fluid inside the cell that kind of holds things up, keeps things suspended. So that's all cells, y'all, all, A-L-L. The reason I'm emphasizing that is there's a few like accessory structures that you might see on a prokaryote, you might see on a eukaryote. So these next two can be on either type of cell, and that's going to be flagella. Some will have some eukaryotes have flagella, some prokaryotes have flagella. Same purpose, motility to move around, but structurally they're a little bit different. But again, long whip-like structures. And then cell wall. Remember back to our intro to micro conversation? Animal cells don't have a cell wall, but fungus does. Protists do, right? Algae do. Well, algae is a type of protist. So protozoans and algae. Yep, exactly. And then I see I skipped over one here. Sorry, y'all. Nucleoid, that's going to be a prokaryotic structure. That's all their chromosome kind of hanging out together. Okay. Any questions on these, y'all? We're going to actually spend some time tomorrow reviewing what some of these structures actually do. Any questions? All right, y'all, have a great rest of your night. Hopefully I'll see some of you in the morning. I'll be posting things probably in the morning, probably between 8 and 9:30. And yeah, it's going to, so with Blackboard, let me actually stop the recording now. Hold on a second.